Hello and welcome to the second half of our DNA unit, which is going to be all about building proteins from DNA, or protein synthesis. So first, the first question we can ask ourselves in this half of the unit is, what is the connection between our genotype, the genes we have, and our phenotype, the physical traits you see? And the answer to that question you can write down is proteins. Okay? So basically, our genes tell us what proteins to build, and then those proteins determine our traits. So you can kind of think of an example might be, you might have the gene for darker colored skin. Well, what's going to happen is that gene is going to tell your body to build melanin, which is the protein, the pigment that's responsible for skin color. So every gene that we have is linked to a protein that gives you a phenotype. Another example, things like you have genes for hormones that dictate things like your mood and all of your secondary sex characteristics. Genes encodes for enzymes that influence things like metabolism. Genes also tell you what proteins to build for structural parts of your body, like the way your muscles and your skin look. However, for proteins to exist, they have to be created by our cells, by our ribosomes. So what we're going to be looking at in this half of the unit is how we go from DNA and how we actually read the DNA to build proteins. So we've been using this word genes all along, and really a gene is just a stretch of DNA that codes for a specific protein. So as a reminder, we know that DNA is found in the nucleus. So I have this fancy picture over here. You can see this red part of this diagram is supposed to represent the nucleus. And you can see my double-stranded piece of DNA. And you can kind of think that if every nucleotide in DNA was one letter, one of your cell's DNAs would fill about 7 phone books of information. And amongst a bunch of DNA that doesn't code for genes, you have about 25,000 genes in that phone book that encodes for about 100,000 different proteins. So we know that somehow we have to read this DNA and create a protein from it. And remember, the organelle that makes proteins are ribosomes. We saw this earlier in the year. And where in the cell are the ribosomes located? They're in the cytoplasm. So we have this issue now that we have the information to make proteins. The DNA is located in the nucleus, but my ribosomes that build proteins are in the cytoplasm. So how do the instructions get from the nucleus to the cytoplasm? Well, we use an intermediate molecule called RNA. So in a sense, DNA and its genes are indirectly telling the ribosome how to make proteins, and RNA is serving as that in-between molecule. So we can look at something called the central dogma, which is, the central dogma is how DNA is turned into protein. So if I use DNA to make more DNA, like right here, that's called replication. If I use DNA to make RNA, that's called transcription. And if I use RNA to then build my protein, we call that process translation. So here's a picture of the central dogma. So you can see here I have a chromosome, and remember, one chromosome has thousands of genes on it. So I have a little mini chromosome, a simplified one that just has 3 genes, so 3 distinct stretches of nucleotides. And if I wanted, maybe this gene here encodes for an enzyme that I need to use. So there's, a couple hundred nucleotides that code for this enzyme. So if I wanted to build that enzyme, build that protein, the first thing that I have to do is turn this gene, this stretch of DNA, into RNA. So here I zoomed in to that gene 1, and this is what the DNA stretch might look like. So the first thing that's going to happen is we're going to get transcription. I'm going to build complementary RNA that's a copy of that DNA. And then I'm going to translate it into a protein. So the ribosome is going to read this RNA and put together the corresponding amino acids to build a protein. So obviously RNA is very important in this process. You can kind of think as RNA as a temporary copy of a gene. So maybe if all all of your DNA is 7 phone books. It's kind of like if you made a handwritten copy of just one page. That's kind of like what RNA does. And this is because DNA can't leave the nucleus, but RNA can. So we make a temporary copy, and we bring that copy to the ribosome in the cytoplasm to build our protein. So let's talk a little bit about RNA. So RNA is also a nucleic acid, just like DNA, but it's very different. First of all, it's only single-stranded. So you can see over here my RNA, it's only one strand of the helix, unlike DNA, which is double-stranded. In place of thymine and T, it uses the base uracil instead. Instead of deoxyribose as a sugar, it uses ribose, hence the R in RNA. And because we're only copying one gene at a time, it's a much shorter molecule. It's only about the length of one gene. So if we were going to summarize the differences, DNA is double-stranded, RNA is single-stranded. DNA uses thymine, RNA uses uracil. DNA has the deoxyribose sugar, RNA has ribose. DNA is a very, very long molecule containing thousands of genes per chromosome, where RNA is shorter, just about the length of one gene. And just as a side note, deoxyribose and ribose are almost an identical sugar. The only difference is, whereas deoxyribose has, if I were to draw the sugar, deoxyribose has a OH group here and a hydrogen here. The only difference with ribose is in ribose, this H is an OH. So very subtle difference. So here, again, it's just a chart summarizing the differences. This chart also includes the functions. So you could see the function of DNA is just to contain the genes and hold our genetic information. But you can see RNA has a ton of different functions, because another difference is there are many different types of RNA where really there's only one type of DNA. Okay, so now let's break down into the types of RNA. Now I'm going to tell you now, there are like 9 or 10 types of RNA that are known to scientists right now, and we're discovering new ones every year. But there's only 3 that we're going to talk about that are going to be important for our class. So kind of the RNA that we've been talking about all along, the one that goes between DNA and protein, is technically called mRNA, where the M stands for messenger, and that name kind of makes sense, because mRNA is the message between DNA and the ribosome, the message that tells the ribosome how to build a protein. So this carries the information from the DNA in the nucleus to the ribosome in the cytoplasm to make a protein. A second type of RNA is tRNA, where the T stands for transfer. And this is kind of like a little vehicle almost. tRNA carries amino acids to the ribosome. So remember, the amino acids are going to be assembled together at the ribosome to build our protein. So the tRNA is kind of like the little trucks that go and find the appropriate amino acid and bring them to the ribosome. And then finally, the last type that we care about is rRNA, where R stands for ribosomal. You might remember in our cell unit, we talked about how ribosome is built of both protein and RNA. So therefore, this is a structural component. It's physically part of the ribosome and makes up it structurally. So here's a picture of the 3 types of RNA. So you can see the mRNA is just literally a copy of the DNA. It's a single-stranded message that the ribosome reads to build the protein. You can see the tRNA has a little spot where it can carry an amino acid. So this is an amino acid. This is the tRNA. It's carrying the amino acid to the ribosome to be put into a protein. And then finally, rRNA is actually part of the ribosome. So this is the machine, the organelle that's going to read the messenger RNA and assemble the protein. Okay, so we're not going to get too far into protein synthesis in this video, but we already know that there are 2 steps. Transcription, where my DNA gene is converted to messenger RNA. I'm going to make a copy, and this happens in the nucleus. And then the second step, translation, is where I go from mRNA and I go to my protein. And this happens in the cytoplasm at the ribosome. And this process requires tRNAs as well, which carry amino acids. So we're going to start with transcription. So here's, again, a little schematic of the whole process. I'm starting with DNA. I'm going to unwind it. I'm going to read the gene, make an RNA copy, and then that RNA copy is going to travel out of the nucleus and be read by the ribosome to build the protein during translation. So transcription, which is what we're going to start with in the next video, is, you'll be happy to know, a much simpler process than replication. We're going to use just one enzyme, RNA polymerase, to copy DNA and to make an RNA copy. And this, again, this name makes sense. Polymerase reminds me of polymer, so RNA polymerase is going to be the enzyme that reads the DNA and builds the RNA polymer.